I want to talk about transcription and translation. I want to talk about how messenger RNA reads the DNA and makes a copy, and then goes out into the cytoplasm and makes proteins according to the pattern that it made off of the DNA. So, here we go. What we're going to say is that this is a cell. I'm going to make a really, really big cell here. And I want to talk about transcription. A scribe in ancient days was a person who wrote things. So when we're transcribing, we're copying. And then I want to talk about translation. I'm going to talk about those two things here. Well, what happens is in the nucleus of a cell is the DNA. And I'm going to kind of put it in a kind of a double helix like we would normally see. But I'm going to show a messenger RNA copying this DNA strand. It may be one way or the other, but I'm just going to say this blue strand is a messenger RNA that is making a mirror image of the original DNA strand. And then what happens is that as this goes out to the cytoplasm, I'm going to enlarge it. I'm going to put a enlargement ray on this thing to make it look bigger so you can see it. But what is so unbelievable is that a straight up copy of the DNA is not good enough. There are actually introns. There are little pieces, little segments that have to be removed before the messenger RNA can become a responsible citizen. The exons are these little pieces right here that get rejoined to make a functional messenger RNA strand. Now, what happens in the cytoplasm is that there is, and it's much, much smaller than this, by the way, but I kind of draw it like a hamburger, but it is a ribosome. And what happens is this messenger RNA strand reads through this ribosome. And according, in comparison to the nucleus, it would be a tiny dot. It would be something little like that, okay? So it's not, this is way out of line. On the messenger RNA, there are 3 member codes on the messenger RNA. And we have a chart that tells start codons and stop codons. So I want you to tell me right now, what is the 3-letter code for the start codon?
AUG.
Okay, now, one of the things that we learned is AUG is always the start codon, so the very first amino acid in any protein is always going to be methionine. Everybody say methionine.
Methionine.
Okay, very good. Okay, so what I want to do is I want to make about a three amino acid protein. So I want you to find me a code for proline. Give me a code for proline? CCU, okay? That'll be proline. CU. Okay, and then let's do lysine. That's one that kind of makes you sleepy or excuse me, that's not, it's tryptophan. Let's do tryptophan. Tryptophan is the one that makes you sleepy when you drink milk at night.
UGG.
UGG is tryptophan. Okay, UGG. Lysine is the one that helps with cold sores on your mouth. Okay, so there are a lot of these different amino acids that have different functions. So we've got a methionine, a proline, and we have a tryptophan right here. Okay, so what's happened is we have a strand that has this code on it. By the way, these are called codons. And I do need to stop. I do need to disassemble this in the end. So I need you to find me a stop codon. Look on the chart and tell me what's the code for one of those.
UGA.
UGA, University of Georgia, Athens. I can always remember that. I love to watch college football. By the way, these are on the blue strand. I'm sorry, it looks like I started drawing them onto the black here. Okay, that'll be a stop codon. That'll help us know when to stop. Okay, in science, UGA does not stand for University of Georgia, Athens. Forget that. But in college football, it does, which is important. That is important. All right, now let's build this chain. So we've seen messenger RNA, which is the strand, and we've seen, by the way, we've seen ribosomal RNA. That is the ribosome itself. And now I'm going to show you a different kind. The last kind of RNA that we're going to learn about is transfer RNA, tRNA. So what happens is this, that there is a transfer RNA that will bring over the methionine, let's just say MET for methionine, and it has an anticodon that matches to the codon. So here's what we learned. We learned that in DNA, that is a rough looking end. In DNA, adenine bonds to thymine, cytosine bonds to guanine. We know that. But in RNA, it's different. So we have to take that into account. Adenine bonds to uracil. There is no thymine in RNA. Cytosine still bonds to guanine. So we need to keep that in mind. We need to keep that in mind. Notice that there are no T's. There are no T's anywhere right here. You got U's. And U's don't match in DNA. Well, help me determine the anticodon that is opposite of this strand. What is the opposite of A, class? U. Okay. What's the opposite of U? A. And what's the opposite of G? Okay. Instead of saying opposite, it might be better for what's the complementary base. I'm just trying to keep it simple. Okay. All right. So that's methionine. And so what would happen then is there would be another amino transfer RNA that would bring in the next amino acid. Well, I think we said this one was going to be proline. So let's say PRL for proline. What would be the complementary base for C?
G.
G. What about C? And what about U? A. Okay, now here's where it kind of starts getting really interesting. This thing works like a machine. Unbelievable. What happens, class, is this methionine, pay attention, this methionine leaves, but it joins to the proline. Okay, I said that wrong. The transfer RNA leaves, the methionine joins to the proline. That's what I meant to say. I said that wrong. The next transfer RNA comes in and it sits down, but this one, I think we said this one was tryptophan. So let's call this one TRP. Help me figure out the complementary basis for tryptophan. A, C, C. Remember, everybody say it. G matches with C. Say that.
G matches with C.
And A matches with U.
A matches with U.
We're talking about RNA. Everybody say RNA.
RNA.
Thank you. Thank you. That's what we're doing. So what happens? This transfer RNA leaves. This proline attaches to this tryptophan. And then look, this is where it all ends. This machine gets down to this point, or this is reading through, however you want to look at it, and it reaches this codon. What's special about that codon? What kind is it? It's a stop codon. So all of a sudden, everything disassembles. This last transfer RNA, let me write that down, leaves. The ribosome disassembles. The messenger RNA is gone. And all of a sudden, we have translated a 3-member protein. We have methionine, proline, and we have tryptophan. So, come back over here to the left here for a second. So, to summarize all of this, messenger RNA copies the DNA. But remember, when it copies it, it doesn't make T's, it makes U's. We have to remember that. It gets modified, it gets spliced. The introns are removed and the exons are rejoined to make a functional messenger RNA. Then this messenger RNA strand reads into the other kind of RNA, which is called ribosomal RNA. Transfer RNA then shuttles over the amino acids so that the amino acid strand can be made. This process is called what, class? That's called translation. It's an unbelievable process. This is like a machine in your body. There is intelligence embedded into this. I'm here to tell you, there's intelligence all in this system. It doesn't work randomly. That's not how it works. You get a random operation here and you get a dysfunctional protein and you get a person that can't function. Okay? Has to be perfect. Listen to this. In humans, if you have just one amino acid defective in a hemoglobin chain, all of a sudden the person has sickle cell anemia. One problem. One. Only one. We're talking about a long chain. So this is a very intricate and detailed process.